genome reconstruction and the woolly mammoth. What if extinction wasn't forever? What if, with a few breakthroughs in genomics, we could rewrite the fate of a species? But the most important thing, should we? For thousands of years, the woolly mammoth has signified a lost world and has been buried beneath layers of ice and time. Yet today, scientists are using advanced genetic tools to ask a question once not thought possible. Could the mammoth walk the earth again? And at the center of that question lies a powerful and complex frontier in genetics, 3D genome reconstruction. Let's start with what most of us picture when we think about bringing back extinct species. Finding the DNA of an extinct species, putting it together, and recreating it like in Jurassic Park. But there's a catch. DNA by itself is not enough to build a living organism. Scientists have sequenced almost the entire mammoth genome. However, the genome sequence is like having an instruction manual in front of you with no clue how the pages are meant to be organized. We know that genes don't work in isolation. They operate in a three-dimensional space, rearranged inside the nucleus, with placement affecting which genes are active and when. Without understanding the 3D structure, we can't truly rebuild a mammoth. We just have fragments of a blueprint, not the living organism. This is where the 3D genome reconstruction comes in. In a 2024 study published in Cell, researchers achieved something extraordinary. Advanced mapping technologies were used to model the 3D structure of the woolly genome. This means scientists can now see how genes folded and interacted inside mammoth cells, how promoters, enhancers, and regulatory elements work together to control traits like fur growth, fat metabolism, and cold resistance. This technology is more than sequencing. You can visualize biology at the molecular level. With these insights, scientists are now editing the DNA of Asian elephants, the mammoth's closest living relatives, using CRISPR to insert the mammoth-like genes into their genomes. There's a company called Colossal Biosciences that is working in collaboration with the conservation group Revive and Restore, trying to create an elephant-mammoth hybrid capable of surviving in Arctic environments. In other words, 3D genome reconstruction isn't about the past, it's about re-engineering the future. But as our science and technology develops, a deeper question emerges. Just because we can bring a species back from extinction, should we? On one hand, bringing back certain organisms could help restore ecosystems. For example, mammoths played a key role in maintaining grasslands that slowed permafrost melting. Reintroducing the mammoths could then, in theory, help combat climate change. However, as biologists have warned in the annual review of animal biosciences, this technology blurs the line between conservation and creation. Are we saving a species or inventing a new one? There are some more questions too. An elephant with mammoth genes inside of it isn't truly a mammoth, it's a living experiment. What responsibilities do we humans have towards that organism? And what happens if it suffers or cannot survive in the wild? Opinions remain divided. Some view this de extinction as an inspiring triumph of science. Others worry it reflects scientific overconfidence, a step towards treating life as something that can be endangered or even commercialized. Ultimately, 3D genome reconstruction gives a breathtaking new lens into the complexity of life. It shows us that genes aren't flat lines of code, that they are living structures shaped by millions of years of evolution. But as we stand at a crossroads of technology and morality, one final question remains. Are we bringing the mammoth back to life? Or are we simply bringing our own ambitions to life through the mammoth? Maybe the real power of this science lies somewhere different from resurrecting the past, but about understanding how fragile and interconnected life truly is, and how carefully we must tread when holding its code in our hands. Thank you. Here are the